Now, my knowledge on the scholarship of life's immediate origins is pretty much limited to some of Richard Dawkins' books, in which he describes a hypothetical process in the primordial ooze and of this world in which molecules, through a process of natural selection, become increasingly complex and develop primitive self-replication abilities, and then these self-replicating proto creatures then develop modes of protection, so primitive cell walls, for instance. And on this goes until something like the early creatures we've just been discussing arose. So how closely does this picture I just described correspond to the origins of life on our planet as you see it? Of course, not entertaining in this case the panspermia hypothesis, and because you already mentioned this a little when you said that the early chemistry wouldn't have happened with oxygen in the environment.
Yeah, yeah. There's actually a very rigorous and vigorous uh, laboratory research effort on so-called prebiotic chemistry, and in a sense, it is, as you've described, a perspective that goes back to the Miller-Urey experiment, which really kicked this whole chapter in scholarship off. And that is what Stanley Miller did was basically took um, ammonia, water, methane, missing something, CO2, and put them in a flask and ran sparks through them to simulate lightning passing through the early atmosphere. And, after this was going for a fairly short time period, he started noticing brown gook accumulating on the sides of his beakers. And when he analyzed that, there were a number of amino acids that had been created. So we know through not only this, but a large body of work under various conditions since then, that it is fairly straightforward to make the basic building blocks of life from simple inorganic precursors under plausible planetary conditions. So you can make amino acids, you can make sugars, you can make lipids. And in fact, people have now succeeded in even making, the materials that make up DNA and things like this. So getting those precursors is fairly simple. Now, it gets more challenging when you start to link them together into functional entities that actually interact with one another, so that, when you replicate your own DNA, you need proteins to do that work. And yet you need nucleic acids to code for the proteins that are doing the work. So how do we get nucleic acids and proteins to work together? And that's still something where there are a lot of unknowns. But one of the things that's kind of fun is an idea that's been around since the 1970s, but seems to have a new life, is that many proteins actually function with the help of what are called cofactors. Some of the vitamins are cofactors, for example. And many of those cofactors actually have a chemistry that it resembles that of, DNA or RNA. And so some people think, and I think it's, you can't prove it, but it's not unreasonable, is that early on, you do, as Dawkins described in your statements, have nucleic acids that can self-replicate. And we do know now that some nucleic acids can actually function like enzymes. We usually think of proteins as enzymes, the things that actually do the work in the cell, but some nucleic acids can do that. And through time, we seem to have, it may be that the intermediate in leading to the world of proteins was some of these cofactors that, maybe that the amino acids of the proteins were originally there to stabilize the cofactors and then became the most important part of it. And so all of these things are possible. Progress is being made on a year by year basis yet, but I think getting that interaction between proteins and nucleic acids that is the basis of modern life, we're not quite there yet. As far as environments, Darwin talked about a warm little pond in a letter from the 1870s, and more people are looking to lakes because lakes can concentrate these organic materials. Lakes can dry up and re-wet, which actually facilitates forming polymers now too. So we actually live at a moment in the history of thinking about the origin of life where there are insights coming from various perspectives, ranging from Earth history to experimental chemistry to insights from molecular biology that are, slowly putting things together. We know a lot more than we did when I was a boy, and I suspect that we'll know a lot more when the boys born today and the girls born today are my age.
Well, I personally prefer another theory, one that I think is much more elegant, parsimonious, beautiful, and that's spontaneous generation, where if you have a bale of hay in just the right barn, eventually mice will spontaneously appear. I much prefer that, but I do understand that it has fallen out of favor. And you've just pointed to a number of theoretical and conceptual tools, I mean, experiments, chemistry, etc., that bridge this gap between the period in which we know that there was no life and these early fossil records. So we have an understanding of how the basic building blocks of life could have formed. Beyond this, are there any sorts of structures or features of the early proto-life that we have a pretty good idea of how they might have formed? So, for instance, I have the understanding that maybe a primitive light sensing mechanism, we can understand how that might have formed, or something like flagella, little ways that tiny creatures can propel themselves about. Are there structures like these that we can just see how they happen through natural selection from these basic or slightly more complex than the basic building blocks?
It's fairly complicated and fairly tough, and I should say before I get into it, with regard to spontaneous generation, several months ago, I took part in a celebration of Louis Pasteur's 200th birthday at the École Normale Supérieure in Paris, and I have to say that old Pasteur was pretty hard on spontaneous generation.
Yeah, well, I can't blame him. I do think it's fun. It's fun to think about. I mean, it's fun to try to put your head in other people's heads when they didn't have the science that we have.
Oh, sure. Yeah, no. I think in the 1600s that made as much sense as anything. And again, it was, you talked earlier about most of the history of thinking about life was a history that didn't include bacteria, and really I think it is because Pasteur was one of the early people to appreciate what bacteria are and what they're doing that allowed him to have these experimental insights with regard to spontaneous generation. But be that as it may, there are certain things that we have some confidence in. So, for example, it is possible to make lipids by non-biological processes. And if you look at, there's a whole class of meteorites called carbonaceous chondrites that can have several percent organic matter in them. And that includes lipids, and they're made,
Fatty meteors,
abiologically. Nice things about
fatty
pardon?
meteors.
Yeah, basically. And if one of the things that we know is if you have lipids in water and you splash the water, they will spontaneously form spherical vesicles. And when you put them in, let's say, a beaker with, amino acids or polymers of amino acids, peptides, and then let it dry out and let it wet again several times, you'll find out that some of those peptides are now actually inside the vesicles. So very simply, the most basic overall features of a cell, which is to have a protective membrane on the outside and the actual chemical work of the cell on the inside, that all makes sense. Precisely how that happened, what the course that takes us from there to a simple bacterium is, there's a lot to learn. And then, when it gets to more complicated things, and a flagellum is actually a fairly complex thing. There's lots of proteins and everything in it. We get some insights from comparative biology. And so, we know, for example, that the mitochondria in your cells originated as free living bacteria that essentially became symbionts inside a host cell. We know that the chloroplasts of an oak tree started as originally free living cyanobacteria that were incorporated and essentially reduced to metabolic slavery within a host. So there are things that we know. And I think once we get into, the last common ancestor of all extant life, which is already a complex organism, from there on, I think we can make reasonable insights from, comparative biology, from molecular biology, that sort of thing. It's getting from the Miller-Urey experiment to the last common ancestor that is exciting, and we're learning a lot, but there's still a lot to learn.